Hi guys. Hello everyone. Today I'm presenting my project on quantifying the effect of glycine rebel switch on the cell fitness of Bacillus subtilis. And my name is Dan Vuong. I'm working in the Meyer lab under Elise, who's been helping me working on this project. So I would like to begin by talking a little bit about the central dogma of biology. So it basically describes the flow of genetic information in biology. On the left, you have DNA, and the information in the DNA is transcribed into RNA. Then through translation, it gets coded into proteins, which are the functional molecules of your body. And throughout the years, an unequal amount of attention has been paid to both ends of this central dogma. But recently, there's a shift. We discovered that RNAs there's a lot more to RNA than just coding. We have ribosomal RNA that facilitates the protein synthesis of ribosomes. We also have RNA, non-coding RNAs that are only present in eukaryotes, and non-coding RNAs that are only present in prokaryotes. And the topic, the type of non-coding RNA that I will be talking about will be the ribose switches. So what is the ribose switch? You're looking at a strain of a graphic representation of an mRNA strain in bacteria. So if you look at the green region to the right, that's the gene itself. That's the region that will get translated. And to the left of it, that's the 5' untranslated region of the mRNA. The right structure is the ribose switch itself. It contains an aptamer domain. An aptamer is basically a sensor for any ligand, for any specific ligand that binds to it. And another feature of a ribose switch will be the expression platform. This works by when the ligand binds to the aptamer, the ribose switch folds differently. That folds either terminate transcription or initiate or inhibit translation. So that's how ribose switches regulates gene expression. The switch that I'm working with is the glycine ribose switch. And this is special because it is a tandem type ribose switch, meaning that we go back to a typical ribose switch, it only has one aptamer, and whereas in the case of the glycine ribose switch, it has two aptamer. Both controls the same expression platform. That's the terminator stem loop. And another key feature is that this ribose switch controls a glycine cleavage gene that is very important for glycine detoxification and transportation. And another notable feature is the leader-linker kink-turn region. During in vivo study, when people did not include the leader-linker kink-turn region, they found that this ribose switch needs to bind glycine to both aptamer, that's cooperativity, in order to work. But after people discovered the leader-linker kink-turn region and include that in their in vitro study, the ribose switch binds independently in that it loses its cooperativity. And this is the mechanism of how a ribose switch function. If you look to the left, this is the ribose switch in the off position. When glycine binds, the two aptamers interact with with each other. That's the gray arrows, and it falls into a conformation that allows for the formation of the anti-terminator, and that's how gene expression can begin. On the right side, that's the 3D structure of the folded up rebel switch. The Meyer lab has worked on the function and activity of the rebel switch in vivo before, and we found that double ligand occupancy is not necessary for rebel switch activity, that is not cooperative in vivo. And also, different mutations affect the rebel switch differently. If you look at this bar graph, the most left group of columns are the wild type, and the rest are mutations. So that it is evident here that the mutations carry different weight when it comes to rebel switch function. Some eliminates the function altogether, while some other you can see elevated function activity. Sorry, pertaining cell fitness. Cell fitness is just a value that we assign to describe a cell's ability to thrive in any particular environment. And because previous data hinted at different growth behavior between the mutants, my project is primarily interested in seeing that whether all these differences add up to the survivability of the mutants. So we use mutant M1, M5, and M6, previously created for this paper, also from the Meyer lab. And the M1 mutation affects the first glycine aptamer. The M5 mutation affects the leader linker kink region, and the M6 affects the terminator's stem loop region. And for each mutant, we have 2 colored strain per se. As you can see here, this is the source plate that I use. I have 4 strains, and for every strain, I have 2 substrains, that is white and blue. And to the left, that's the only difference between the strain, and that one can break down X-gal to give a blue color, and one cannot. For the assays that I use, I typically mix 1 blue and 1 white strain together and culture them overnight, either in the presence or absence of glycine. I would plate the starter culture and also patch plate on X-gal plates. That's something I'll go over later. And for the cultures, that's the starter plate. And for the plus and minus glycine plates, I would culture them. I would culture it overnight and also patch plate that as well. The long-term experiment that I use is very similar. The only difference is that I continue culturing, serial culturing, over the span of a few days instead of 1 day. This is what a typical patch plate will look like. I try to patch over 100 colony per plate. You're looking at this example plate is a mixture of M5 blue and wild type white, and that is cultured in the presence of glycine, and that's the second day of a long-term experiment. And to the right is what my bench looks like after a week of experiment. So yeah, for calculating the fitness, this is the formula that I used, and this basically accounts for the starting and ending ratio and the expansion factor, which is how many times the culture has doubled per se. Result-wise, this is the numbers that I got for the short-term experiment, and I will be going into this by strain. If we look at the wild type, to the left, that's the in vivo activity study, and we can see that in the absence of glycine, that is the white column, the activity is very close to 0, and with the presence of glycine, the switch turns on as expected. We expected to see a fitness of 1 for wild type, because we're just basically comparing to strain of the same genotype. But as evident in the short-term and long-term data, we saw some noise. The fitness is, well, not very close to 1, but for the long-term experiment, we started with a 1-to-1 ratio of wild type to wild type, and we ended at something very close to 1-to-1, but you know, you can see fluctuation in between. For M1 sees the least decrease in fitness, but one thing interesting is that if you look at the in vivo activity of the switch, M1 greatly decreases in activity. The fold is about 12-fold decrease in activity, but if we look at the fitness, it did not decrease by much at all. Still, you know, close to 1 relative to other strains that I'll be showing you. For the long-term experiment, we see the effect of the mutation more clearly. We started out with a 9-to-1 ratio of mutant per wild type, and throughout the span of the experiment, the ratio has decreased, meaning that the wild type was starting to outcompete the mutant. For M6, this is also interesting, because if we look at the in vivo activity of the glycine rebel switch, it was actually upregulated compared to the wild type. But when we look at the fitness, we see a decrease in fitness compared to the wild type. And long-term experiment makes it even more apparent. And that if you look at the solid blue line, that's the percent of population in mixture that is M6. You see about a 50% decrease in percent throughout the span of the experiment. If you look at M5, it's even more unfit. The short-term data by itself shows that M5 is a lot less fit than the other strain, in that the fitness value are significantly lower than 1 as compared to the results for other strains. And if you look at the long-term experiment, by the end, we started with a 9-to-1 ratio, and by the end, the mutant was completely outnumbered, either in the absence or presence of glycine. So I think what I can conclude from the data that I have is that all the mutations that were involved in the project were not favorable in terms of cell fitness, and the effect of glycine is can be seen more easily in the long term. And one more interesting thing is that the in vivo activity of glycine rebel switch and cell fitness does not always go together. So this is a graphic summation of my data. If you look at M5, you see a very sharp decrease in vivo activity and also a very sharp decrease in cell fitness. But for the case of M1, the in vivo activity decreases by a lot, but fitness only decreased by a little. And for M6, the in vivo activity actually increased, while the cell fitness decreased. So that's what I found very interesting. And as for this is what I wish I could do if I had more time. I would like to sequence the mutants to know exactly what I'm working with, just because the data has considerable amounts of noise and inconsistency. I mean, I know what I'm working with, but I just want to make sure. And I would also like to compete a larger number of mutants to generate a bigger fitness landscape. I would also like to compete the strains under lower or higher glycine concentration and for a longer period of time, because glycine in high concentration is toxic, but I would like to put a value to that as to how much that affects the fitness of the cells. And then I would also like to probe for potential interaction of the gene that the glycine rebel switch controls outside of the glycine cleavage system, because I ran a literature, a database search, and is also it turned out that that gene also interacts with other genes as well. So with that, I would like to thank the NSF for funding this opportunity, and Boston College for hosting the 12 of us. I would also like to thank Dr. Birch, Amanda, and Nancy for making this happen. I would like to thank the Meyer Lab, especially Dr. Meyer, for believing in me when I was still an applicant, and Elise for helping me completing, working through this project. Lastly, my mentors, friends, and family who supported me along the way. All right, any questions? Marissa? Yeah.
So, you mentioned, you made some strains that were blue and some that were white. What was the purpose of making?
Oh, so you can't actually just differentiate by the shape of the colony. They all look the same. And we want to be able to tell which strain, this is like color coding. Yeah. Yeah. Yeah, so that, because we're mixing two strains together, a mutant and a wild type. And we want to be able to tell which colony is mutant and which colony is wild type. Yeah.
Can you go back to the meeting? So this is a bit of an elaborate question, but how has the summer changed your perspective on the central dogma?
I'm more exposed to the functions of RNA now. To be honest, I did not know about rebel switches before I started this project, so it is very eye-opening. And there's a thing going around biologists that about RNA world, that the origin of life starts with RNA and not DNA, but that's still debatable. Yep. Melissa?
Can you go back to the slide that had the plates?
Yes.
That. So if I'm understanding correctly, like each circle is a different colony of bacteria?
Yes.
So my question is how did you deal with the possibility of contamination?
Oh, contamination?
Yeah, like if you're growing two different colonies.
Yeah, these are all grown on antibiotics plates.
No, I mean like when I mean contamination, I mean how did you find a way to make sure the two colonies would grow together?
Oh, actually, they grow together. So if you look at this is, so I plate them on normal plates.
No, I mean like.
Oh, I just touch one colony with a toothpick and then touch it to an X-gal plate. And so, for example, imagine if you plate like a mixture of bacteria, it will grow randomly on the plate.
Right.
Then that's the purpose of patch plating, in that you put them in order. Yeah, it's elbow grease. So, anyone else?
Okay, let's think.